Assalamualaikum. I'm here with FSc Chapter 3 Enzymes Complete Summary. Small chapter and big impression we should learn to save time. We shall discuss enzyme, its characteristic, its mechanism of action, factors and inhibitors. Enzymes are proteins which increase efficiency of biochemical reaction. They are made of amino acids. They increase rate of chemical reaction or catalyze the chemical reaction, also called as biocatalyst. Enzymes have globular structure and are very specific for the reaction. One type of enzyme has one structure for that reaction. Small part of enzymes which actively participate in reaction is called as active site. Very important to remember. Active site consists of few amino acids, very small number of amino acids, and rest maintain the globular structure. So the question arises, the function of active site in rest of enzyme is to maintain the globular structure. Non-protein part of enzyme, I repeat, non-protein part of enzyme is called as cofactor, which is required for enzyme. Cofactor act as bridge between enzyme and substrate, so it helps to recognize, bind and attach the enzyme with the substrate and then start the reaction. Cofactors provide chemical energy in few reactions. Very important MCQs to remember. Now the example of cofactors are magnesium, iron, copper, zinc, iron, etc. These are inorganic ions which act as cofactors. If these cofactors are detachable, then they are called activators. Activators are not enzymes, these are cofactors which can be detached with enzyme. If it is covalently bonded, it is prosthetic group, and if it is loosely bonded, it is called coenzyme. Coenzymes closely relate with vitamins. How they relate with vitamins? Raw material for coenzyme synthesis is provided by vitamins. Coenzymes are needed in very small amount because they are recycled, means they can be used again and again. So if a small amount is present, it will be enough for a large amount of reactions or recycling. Enzyme without its coenzyme or prosthetic group is called apoenzyme. So that is very important fact to remember, that apoenzyme is the only enzyme which don't have its coenzyme or prosthetic group. This is most important MCQ in this slide. Apoenzymes are usually inactive. An enzyme with its cofactor attached is active and called as haloenzyme. Please remember, apoenzyme are inactive and haloenzyme are active. Enzyme involved in photosynthesis are found in chloroplast, involved in cellular respiration are found in mitochondria, and the enzyme involved in protein synthesis are found in ribosomes. Characteristics. Enzymes are very specific in their actions. They are sensitive to even a minor change in pH, temperature, and substrate. Enzymes lower the activation energy of reaction. Activation energy, the energy required to start a reaction. So if the activation energy is lower, it is very easy to start the reaction. Pepsin. Please remember, pepsin is a powerful protein digesting enzyme. It is very important MCQ that protein digesting enzyme, which is the powerful protein digesting enzyme, pepsin. It is produced in an inactive form to stop it from damaging cells. Internal inactive form of pepsin is pepsinogen. Enzymes have three-dimensional globular shapes. Enzyme can be used again and again. These are very important facts to remember about enzymes and already been asked in ppsc exams. Now the mechanism of enzyme action. Enzymes act on their specific substrate and convert them into products, so substrate change into product. Respiration and photosynthesis are metabolic pathways, not single reaction. This can be asked in a way that which are not single reactions, that are respiration and photosynthesis. Enzymes work in series of reactions when they are asked and hand over their product to next one. So, in a series, the first substrate is converted into a first product or intermediate product, then it is hand over the product to the next one, which makes the original product. In this way, a series of reactions occur and they are called as pathways or cascade reaction. In feedback inhibition, this inhibition occurs when final product of pathway act as inhibitor and stop the first enzyme to start the pathway of reaction. Feedback means return. So what happens in this way? The final product of the pathway act as inhibitor and attach with the start of the enzyme and then start the reaction. Active site is charged, may be positive or negative. Mostly active site is negatively charged. There are two definite regions in active site, binding and catalytic sites. Binding region and catalytic region. Binding region or binding site help to recognize and bind with substrate to form enzyme substrate complex. Remember these two facts, recognize and bind. Now catalytic site then catalyze the reaction and transform substrate to product. These two facts are very important to remember, binding and catalytic sites. Enzymes require aqueous medium water for their activities, so without water enzyme cannot work. In mitochondria and chloroplast, enzymes remain attached with membrane. It seems unimportant, but it can be asked that enzymes where are found, or in mitochondria and chloroplast which functions are performed. Emil Fischer in 1890 suggested lock and key model. This model suggests that active site is rigid structure with no flexibility and is used as template. Coastland in 1959 proposed induced fit model. According to this, substrate induced changes in enzyme structure which enables it to perform effectively. In lock and key model, the active site is a rigid structure and cannot be changed, but in the induced fit model, active site is not a rigid structure, it can be changed. Factors. Enzymes are specific due to their specific chemistry, specific configuration, and their specific structure. If enzyme concentration is doubled, the rate of reaction will be doubled in excess substrate. The condition is excess substrate. So if the substrate is doubled and then enzyme concentration is increased, the rate of reaction will also increase. If all the active sites are occupied, then there will be no increase in rate of reaction. It is because the active sites are the region where the reaction is carried. Increase in temperature increase the rate of reaction up to a certain limit. The enzyme work best at their optimum temperature. Human body temperature is 37 degrees centigrade, which is optimum for human enzymes. Heat provide activation energy and kinetic energy. Very important to note, activation and kinetic energy. Heat temperature denature the globular structure of enzyme. Change in optimum pH change the isolation of amino acid at active site. Extreme change in pH cause breaks the bond in enzyme and they denature. Now optimum pH of enzymes. Enzyme and optimum pH is shown in the table. You can note and remember the optimum pH of various enzymes in this table. Now inhibitors. Chemical substances which react or bind with enzyme and block the active site to work temporarily or permanently are called inhibitors. There are two types, irreversible and reversible. Irreversible inhibitors block the active site by forming covalent bonds, then destroying the globular shape or physically block active site. Reversible form weak linkage with enzymes. They can be neutralized. There are two types of reversible inhibitors, which may be competitive inhibitors and non-competitive inhibitors. Competitive inhibitors have similar structure which have mislead active site to bind them. Non-competitive inhibitors form enzyme inhibitor complex other than active site. They alter the enzyme structure and stop the catalysis even substrate bind with active site. Now, very important fact to remember, malonic acid is competitive inhibitor for succinic acid. Succinic acid is a substance and the chemical structure of succinic acid and malonic acid is similar except one hydrogen. So, malonic acid can attach with the enzyme or mislead the enzyme and can act as competitive inhibitors for succinic acid. I hope you have learned what you needed. Next chapter will inshallah chapter 4, the cell. Please support and stay connected. Thank you. Allah Hafiz.